Hi, and welcome to today's Oxford Centre for Personalised Medicine Flash Interview vlog, where we talk to different people involved in the field of personalised medicine in one way or another. Today, we're joined by a very special guest, Ewan Birney, who is the Deputy Director General of the European Molecular Biology Laboratory and Director of the laboratory's European Bioinformatics Institute in the UK, among many other prestigious roles. Ewan, I can't tell you how much of an honour it is to chat to you today. Could you kick off by giving us an overview of your background, so who you are and how you got to where you are now?
Yeah, so I'm very lucky in that I I did my science just before the Human Genome Project was first brought together in draft form, and I've been in genomics ever since. So I was involved in the initial analysis of the draft genome, in particular around the gene set with my colleagues, Michelle Clamp and Tim Hubbard, starting the Ensemble project. And I have stayed with genomics and with databases ever since. So looking after the human genome, analyzing the mouse, the chicken, and other genomes. I then had a very productive period looking at the non-coding part of the human genome, that's the ENCODE project. And during all of this time, aiming to not only do the analysis, but produce databases with my colleagues here at EMBL-EBI, where I started in 2000 as a team leader. I'm now the director of EMBL-EBI, and I'm Deputy Director General of EMBL, the European Molecular Biology Laboratory, which is Europe's only international organization focused on life sciences. We're headquartered in Heidelberg, Germany, and I run its second biggest site, which is the EBI here in the UK. And the mission there for the EBI is to provide the human genome and other resources like the human genome, so other genomes, but also things like the genes and its expressions, protein structures, the pathways, and the literature to the world. So we take data from around the world. Molecular biology has this wonderful tradition that all of our data is shared on publication. So when scientists publish, they are encouraged, often mandated, to deposit their data in international databases, and we run the European side of those international databases. But in addition to that, we look at all of those data sets and we transform them into more digestible products. And the human genome is a great example of that, where it's about, you looked at all the different experiments that go behind the current understanding of the human genome, it would probably be over 100,000 such experiments. Now, in theory, each researcher could download each individual experiment and analyze each experiment themselves to be reassured that everything hung together. But that would be exhausting for everybody to do. And so what people do is they take very often the gene set or the reference genome from my colleagues here in Ensemble and use that. The same story is here for example, protein structures or protein pathways. I'm sure we could get on to other questions there about the future of AI in this world, but maybe that's enough to say. Let me just add one more thing for personalized medicine. It's been a passion for mine. So I do research here and deliver these research products, but obviously for it to be used inside of healthcare, it's a new technology for healthcare, genomics. I make a strong analogy here with imaging technology. So just like X-rays was a new imaging technology and healthcare had to work out how to use it inside of healthcare, genomics is a new technology for healthcare. Well, not that new, but pretty new. And it has to be internalized and used inside of healthcare. I'm very proud of our role of delivering this, but it's a role that's kind of ongoing in providing this data infrastructure. I'm a non-executive director of Genomics England because I have a passion for this, to see this actually end up in the hands of clinicians and ultimately of patients. But not only just the UK, but I also advise Genomes Denmark. I hang out with a lot of Finnish people in this area, Estonians, cheerlead with the French and the Spanish. And in each case, the healthcare systems, which are very, very varied across Europe and across the world, need to internalize this technology, work out how they use it, to help patients. And of course, genomics, the sort of fundamentals of human biology are the same around the world, but the way healthcare is delivered is really quite varied. So that process of how one internalizes the technology is different as you move country to country.
Brilliant, thank you. So thinking a bit more about the work that you did on the Human Genome Project then, how revolutionary was that project at the time? And how much has the field changed since the original project?
Yeah, so the project conceptually got started in the mid-80s when I was absolutely a kid. So I came only sort of in the middle to the end of it. And it was, you know, it was a real ambition to do the human genome. And the human genome is large. It's 3 billion base pairs. It's not the largest genome around, but it's very large. And we could only determine about 500 bases at a time, or 1,000 if you really stretched it. And so it was a real challenge to even imagine writing down that project in the mid-80s. But visionary people, in particular here in the UK, John Solston, wrote that down and said, look, we think this is doable. We'll obviously have to improve the technology. Will obviously involve effort and technology development to get there. And when the first draft human genome was done in 2000, that was kind of epic. It was done by these kind of almost factory-like processes of teams working using this technology at the time. Now, since then, we've gone through at least one, if not two rounds of technology improvement. So rather remarkably, these days, the equivalent of that draft human genome in 2000 can definitely be done in 24 hours now and better. I mean, it's a bit spooky, to be honest. And the price has gone from the multi-millions, multi-hundreds of millions to do to, you know, thousands or the order of thousands. And then one has to, you know, decide how one charges, or depends how one costs up the informatics and other components. But it's absolutely dropped in cost. So that, of course, means that researchers have been able to use this technology in many, many more new and exciting ways. So there's a big increase around research here. There are new ambitious projects like the Tree of Life project, the Earth Biogenome Project, to sequence every species on the Earth, which, you know, sounds pretty awesome. But of course, that also enables many places to internalize this technology into medicine, make it routine inside of medicine.
Okay, so do you feel like the ambitions of the project have been fulfilled then in medicine? And what do you think would be the next big step forward?
Yeah, so I think, so genomics is going to be a kind of evergreen project about how we use it. The ambitions of the project are hard. I mean, at a very basic level, the project was to determine the human genome sequence, and that was definitely fulfilled. We had always had a little asterisk on that it was the bits that we could easily determine. And for the bits that we could easily determine, that got finished in 2003. Now, as it happens, there was a little asterisk, there's a little piece of small print in 2003, like these parts of the genome are really, really tricky and they're really annoying, and we don't think we're going to get around to them for a long time. That recently, just in 2022, 2021, 2022 fell because of this long read technologies, which has allowed us to get through the last bits of the human genome, the centromeres, and in particular, the ribosomal repeats. So that is awesome to see. Actually, it's kind of amazing. But that's the project, even back in 2003, was transformative for research. And you immediately ask the next questions about how do we use it in different ways. And here, I think the analogy with X-rays is useful, because actually Röntgen, when he first saw his skeleton, his hand in the X-ray machine from the X-rays, and he saw his wedding ring, the next day he wrote, this will have medical applications. He's a physicist, he's playing around with radioactivity, and he says this will have medical applications because you can see inside the body. But it takes 30 years. It's quite a long, complicated story about how X-rays become not merely safe. There's a whole business about them being safe and safely used, and the machines being effective and easy and deployable, but also for medicine to work out how you use them. And I think that story is a nice story to go back to. And it's a nice analogy because everybody in medicine, uses imaging when appropriate for different things. There's different modalities, there's different ways of using it. Most clinicians have a feel for when they should get an image done and what they should expect to see in a record about a particular diagnosis. Have you done an X-ray? Have you done an MRI? Let me have a look at the MRI scan. And I think we're going to end up in the same place with genomics. So it's going to be one of these technologies that for certain things, we will just say, look, we've got to look at this guy's genome. And then, of course, there's other areas where the genome won't tell us so much. So has the ambition been fulfilled? It's a tough question to ask in the very broad sense, because I just think it's ongoing. It's a routine innovation development rollout process, just like imaging from Rotterdam onwards has been a slow innovation and absorption into medicine.
I also hear that you organised a rather fun sweepstake in the genetics community back in the time of the project. Could you tell us a bit more about that?
So it was good. It was great fun. So I'll take you back to the late 1990s, the dot-com boom. The human genome is being put together. The community comes together, still does come together once a year in Cold Spring Harbor in Long Island, New York. And classic scientific conference, you're kind of showing off and you're listening to your colleagues. And a big discussion was how many genes are there in the human genome? And one context here was that there was a kind of on the cheap way of doing it, which was called EST sequencing, and a company called Insight had done this, and they were keeping it private, and they were selling access to 100,000 human genes. So that was one marker on the table. And other people had done some calculations, which were pretty noddy, which was like, this is the average size of a gene. This is how big the human genome is. I'll just divide the average size of the gene by the human genome, assuming everything's packed at the same level. And you end up with sort of 70,000 or this sort of number. Though, of course, that makes an assumption that the density is the same and there isn't any gaps, which we now know to be false. And there was a particularly brave and bold Frenchman, Hugues Roest-Crelius, who used an evolutionary technique. And they took a fish genome, which they did a shotgun skim of a fish genome, and they did some comparisons, and they came up with the shocking, at the time, 26,000 protein coding genes. So it was in the air. Here were 3 data points. And so I said that in my presentation, where we also were looking at where genes were in the human genome, this is with the Ensemble Project, with Michelle Clamp and Tim Hubbard, and the other people in the project. I ended the presentation saying, and I will be accepting bets on a sweepstake for the number of human genes. And I said, this year it will be $1 a bet, and next year we'll know more, so it'll be $10 a bet, and the year after, $5 a bet. And the year after, it will be $20 a bet. And it's a sweepstake, you pick a number, the closest number wins. So I went around that evening, around the bar. I basically got nearly everybody to place a bet for $1. So there's an awful lot of bets. And let me now fast forward to 2 years later, where the bet is meant to be called. 2 things happen. So the first thing is it becomes absolutely clear that everyone who betted too high. No one betted low enough. So although we couldn't we didn't know the precise number of protein coding genes, and there's quite a fiddly gray area of definitions and other things. And the betting book has actually quite an interesting, for the purposes of the bet, this is the definition of a protein coding gene. Although we couldn't come to that final number, we knew who won because we knew who had the lowest bet. And at the time, so that was Lee Rowan from Washington State, and Hugues Rost-Crelius, the Frenchman, still works in Paris at ENS. He actually had the third lowest bet. And so we decided that we should give it to the sort of bottom three, which I thought was very fair, because I thought the only Lee, maybe she did come to that from her own perspective, but I absolutely think that the bet was anchored at the bottom by Hugues, who kind of stuck to his guns. Now, if you go back into the literature and you read the genetics literature in the 1970s, they come up with a gene number that's much closer to 20,000 protein coding genes, interestingly enough, by pre-molecular biology genetics. So this story is a fun story. It's a story of how, when scientists don't have data, we can be wildly wrong as a community. Yeah, it's a little bit of history.
Fabulous. So if we fast forward a bit to today, then, what's the main focus of your research now?
So there's two ways of saying this. So let me just stress the sort of strategy side of me, running the EBI and advocating for genomic medicine across Europe and around the world. And there's a really important mission here to ensure that these databases stay up to date, stay relevant, and move with the technology, and are the bedrock. So everything we do in science, although we publish papers in biology, really the final repository of our understanding are in databases. So it's like the modern Library of Alexandria here, and it needs to be kept. It needs to be kept up to date. It needs to be kept relevant. I often make an analogy with the electricity grid. So you and I, neither of us woke up this morning and thought, I better ring up the electricity provider in wherever we are, Oxford and Cambridge, and check that electricity is going to be running. We walked out of the door assuming that electricity will be working, and we do that every day. And researchers, molecular biology researchers around the world, and now clinicians, write grants, do analysis, do experiments on the assumption that the human genome information will be available tomorrow, in a week's time, in a year's time, in a decade's time. And not only will it be available, but it will be up to date and the best as it can be. All of our understanding will be integrated into that sort of artifact. And it's really important that we execute that. And so I work here with actually around 600 people. Believe you me, I don't. I do PowerPoint presentations and vlogs rather than doing it. And some great colleagues here at EBI delivering those data services to the world. I still run my own research group, and if you indulge me, I can talk a little bit about my own personal research. So I have two organisms which I really love to study. So one are humans. Humans are really interesting. So we are obviously mammals. We are pretty standard mammals when you get down to it, and we've got a big brain, but otherwise we come with a pretty standard mammalian organs and systems. And because of medicine and because of the importance of human health, we track millions of humans around the world. Very often we sequence their genomes and we do the genetics, and there are these amazing systems from medicine for measuring things of humans. So imaging is the really obvious one. We measure lots of things through imaging, but also like very simple things like height and weight and all of that sort of stuff. And then finally, of course, when people interact with hospitals, these days in many hospitals, that leaves an electronic trace of that interaction in the system. And so as researchers, we can access that, and you can access that in a variety of ways. So one way is there's these dedicated research cohorts. One of the best ones is here in the UK Biobank. Your colleagues in Oxford are big in that, and up in Manchester. And that's 500,000 Brits going around their everyday business, and they have responded to an advert saying, would you volunteer to provide your information anonymously for science? And they've said yes, and it's great. And that started 20 years ago, and we've been tracking that cohort for a long time. Actually, the UK has a really good history of these cohorts, and there's a cohort, for example, that started in Edinburgh in 1936, which is still being tracked today. So there's a good tradition of that. I also like Denmark. Denmark is a particularly good place for doing this because back in the 1960s, they started to do the electronic healthcare records for certain diseases. And basically in the 1970s, this became far more routine. In the 1990s, it really went full on. And so you can, as long as you go through the ethics board and you ask the research question that you ask is well designed and well focused and is approved, you can get access to these healthcare data with very often genetic data and do studies. So humans are great, and I focus more on understanding human biology than necessarily understanding human disease. A couple of reasons. One reason is that human disease is a very populated research area. And secondly, actually human biology and healthy humans is kind of the inverse that we have to understand disease. If you don't understand how it works normally, it's very, very hard to understand when it works abnormally. But there's lots of things you can't really do in humans, and in particular, you can't tell humans how to live their lives. So you can't say, look, for you, I'd really like you just to not eat so much for this. So that all of that business of how humans interact with the environment is really tough to get at in these human studies. You can get at it a bit, but it's really, really tough. And so here, my second favorite species is, would you believe, Japanese rice paddy fish, Medaka fish. They are beautiful little tiny fish. And as the name suggests, they live in Japan, in rice paddies, also in Korea. You might now ask yourself, why Japanese rice paddy fish? Why not mice? Why not rats? Why not something else? So it happens to be, and we don't really understand why, that Japanese rice paddy fish, we can inbreed in the lab. So we can take wild individuals and we brother systemate them, and most of the time they don't die. Now, this is not true for other vertebrates. Most other vertebrates won't inbreed to homozygosity in the same way. And Medaka fish will at a much, much higher rate. And because of that, what we've done is we've taken sort of a little rice paddy, it's quite a big rice paddy in Japan, and we've captured some fish from there, and then we've inbred them. And it's like taking a little town of humans and now freezing that genetics, so I can reproduce the genetics of that little town again and again and again and again. And if you're a clinician, especially a cardiovascular or you're a cardiovascular researcher, you'll know this famous Framingham study in Boston, Massachusetts, where they went to a town and they interviewed everyone back in the 1950s. It was a real bedrock for cardiovascular research. And so it's like we've got the Framingham of Medaka, but we can now reproduce Framingham in many settings. And so we can move Framingham from Boston to somewhere else to the south. We can ask all of Framingham to the Medaka Framingham to stress, don't eat or do eat, or we'll expose you to this or expose you to that. So it gives us a lot more experimental control of this vertebrate, but still in a setting where the genetics are wild-like genetics. And so that's why we look at Medaka fish.
So bringing the conversation more towards the aims of the CPM, I know we spoke about it a little bit at the start, but what does personalised medicine actually mean to you?
Yeah, so I think it's a very broad statement, and it means different things to different people, and that's fine. I actually prefer the phrase genomic medicine for the area that I'm in because it's more specific. For the people, just to stress, I use genomics here to mean genome-wide assays, so not just germline, but somatic and transcriptomics and proteomics and all of that stuff. It's just really clumsy to say multi-omic medicine. It would be a really clumsy phrase. And I think all of these things, just to sort of take it back up to personalized medicine, is we're going to take measurements of individual humans, including their genome, and then we're going to combine those measurements for working out what we do in terms of clinical decisions for that individual. And a big success story has been rare disease. And I don't know, should I talk about that now? The rare disease story is great. Yeah, so I mean, since the 90s, since the discovery of the ability to do routine genetics, we've always thought about these genetic diseases, which individually can be very rare. Collectively, they're not so rare. So it's about 2% of live births have a child which within usually the first 6 months, they will present with a concern to the pediatrician. Pediatrician will say, I think there's something congenitally wrong with this child. Very often it's about facial features. And what has happened is that in the 1990s, there was a rather laborious process of guessing precisely which gene was involved, and you guessed, and then you tested the child, sometimes the parents, for that gene. And then if you guessed wrong, you would try another gene and you would go along in that route. These days, the far better way of doing it is you just sequence the entire genome of the child and of the parents, so the trio. And when you do that, depending on the disease, about 20% to 30% of the time, the geneticist can make a definitive diagnosis. So you can say, the reason why this child has these presenting concerns is due to this gene. Now, what that does for nearly all the children is, when the diagnosis happens, is it really improves the management of the ongoing concern or disease. And that has been shown by a recent paper where the number of hospital visits actually halves post-diagnosis when people have looked at that. So that's good for the child. It's better care for the child. It's also good for the NHS or for the healthcare system. Very occasionally, there is a transformative treatment for the child. And there's one which is looking at, for example, a gene which causes blindness. When there's a defect in the gene, the child will almost certainly go blind. And now there's a gene therapy that works very effectively. And so you inject a virus that has a good copy of the gene into the retina of the child. The virus integrates that bit of DNA into the right cells, and the child doesn't go blind, which is pretty good. There's another one which is about when the child's immune system simply doesn't develop, or doesn't develop anywhere near as well as it should do. And so there you give the child a bone marrow transplant. And there was a very good example here where a child came in with like chicken pox multiple times and different infections, and the child's about 6, and they sent the genome and the parent's genome for genomic sequencing, and they discovered that in fact, indeed, the immune system, the child had one of these genetically defective immune systems, and so they performed a bone marrow transplant with that child. So over time, and the final benefit of all of this, so we talked about the child in general, we talked about these individual cases, but then also it is really transformative for the parents in 2 ways. So many parents feel a mixture of a responsibility to really do the best for the child. Of course you would. And so by getting a diagnosis, it brings closure. And that's the word often used. It brings closure to those parents, and they say, I now understand what's going on. And a sense of relief, I think, for some parents, that's definitely been documented. And the second thing for those parents is if they would like to have another child, when a diagnosis happens, they can get much better advice. So very often these genetic diseases have happened only in that generation. So they're not present in the parents, and it emerged just in the child. But without a diagnosis, this other situation can also happen where there's a recessive gene, both parents are carriers, and the child it's a 1 in 4 chance for the disease to emerge. And without a diagnosis, you can't distinguish those two examples. Now, in the case where it's de novo, it's probably a 1 in 1,000 sort of chance of it happening again. In the case when it's recessive, you can then, parents can be advised that they could take up pre-implantation genetic diagnosis. And that's where IVF happens, and the children, the embryos are then tested for whether they have the disease, and only the healthy embryos are implanted into the mother. And that's been a process that's very well regulated in the UK and has been running since the 90s, and that gives those parents another option. So you can see this is just one of these kind of crazy win-win-win-win situations. Like everybody wins. The healthcare service wins because it's less costly than the previous way of doing it, and there's less visits by the children who get a diagnosis. The children who get a diagnosis get a win because they get better care. Sometimes it's transformative. Usually it's just a little bit better, as it will, or it's better management. And then the parents get a win. And this is only going to improve in the future. We should be doing this everywhere in the world. We should do it routinely everywhere in the world. All the children that have these presenting concerns should go through this process. And I have a passion to try and make that happen in all the different places. I am very, the UK has a very good program here with the Genomics medicine service in the NHS coupled to Genomics England in the UK, and then I'm going to get it wrong, Scottish Genome Partnership in Scotland to do it. The Scots are very good at this. So yeah, the UK does it very well, and I would like that to happen in more and more places around the world.
I also believe that you're quite a key proponent for responsible data sharing and creating standards in genomics with the Global Alliance for Genomics and Health. So why is this so important for personalized medicine?
Yeah, so there's two sides of that. So one is what I just mentioned, the Library of Alexandria at the EBI, that kind of baseline fundamental knowledge. You can't, English text is not the way you transmit this knowledge at scale, or even just in details. You just can't write it. I mean, you can write it down, but to use it, it must be inside of computers. And that means standards and development. And that's a big part of what the EBI provides today. And it will continue to, I think, be a big part of the future, forever, basically. We have this extra interesting thing about humans where we are tracking and measuring and caring for so many humans around the world through healthcare. And it is a great opportunity for human disease research, for healthcare research, for making all of us healthier, but also for just understanding biology as well. So there's also this kind of side effect on just understanding how biology works. But to access that, we need responsible ways of researchers getting access to this, often at scale, and that needs to have the trust of patients, citizens, and governments involved. And GA4GH sets up that side of it, which is how do researchers get responsible access to data to do health research in a way that patients, citizens, governments, healthcare systems can trust that this is happening appropriately. And you can imagine that there's technical sides of this, like how do you actually do the computer side of it? And that's actually quite involved, and it's quite fiddly and complicated and technical. But there's also something which is much more understandable by a broad range of people, like who makes a decision about what research happens? How does this work? Who gets to decide all of this? And that is a sort of societal, moral healthcare problem. And we root this discussion in the UN Declaration of Human Rights, which has two human rights here. One is the right to benefit from science, so the right to science, to benefit from science. And the other one is the right to participate, the right to participate in science and the right to contribute. Now, these are not well-known human rights, but they are in the UN Charter of Human Rights, and many, many countries have signed up to those human rights. So that is the starting place for the conversation that ultimately has to happen in a national system. And if you indulge me a little longer here, just to stress the conversation you have in Denmark, where they say, well, this makes total sense. We'll go to the Danish parliament and we'll have a piece of legislation about how this works. That's the way the Danes do it. The conversation you have in the UK, for example, involves the NHS. What people trust is the NHS, and sort of don't trust the government quite the same way as they trust the NHS. So for the citizens and patients in the UK, they're like, I would like to see an NHS body in charge. And I would like to know that at the top of that, there are clinicians who have the NHS, people who have the health of the nation in their mind when they make these decisions. And if you go to America, it's often patient advocacy groups. So it's the people who are campaigning for like, let's solve this disease or solve that disease. That's the place where a lot of people will put Trump. Now, all of these are valid. None of these are better than the others. They're just different ways of wiring this together. One can understand why different countries come to different decisions. The thing to realize is that there isn't one way to do this. It has to be appropriate for the society, the way that society is structured. And that's what GO4GH helps realize and then deliver into a technical framework.
Perfect. I won't keep you for much longer, but just two final quick questions. So where do you see the field of personalised medicine going in the next, say, 10 years? And what are the big challenges that we need to overcome to get there?
So one obvious area is cancer. So cancer is very often, these are cells that have gone amok. They are replicating without control inside of our body and causing all sorts of problems. And very often we can characterize cancer by taking images of them down a microscope. That's called pathology, histopathology. And we can also characterize them by their genomic characteristics. And both of these come together to understand why this particular group of cells are in this particular person has gone completely haywire. Now, 1 in 2 of us will sadly get a cancer diagnosis at some point in our life. And when we can make better decisions about which drugs work for which cancers in which people, it's just a benefit to everyone. Now, this is hard. Cancer is very heterogeneous and very complicated. It's like everything is complicated. And also you have to make decisions really, really quickly. Can't hang around. So it's a great opportunity. Genomics is being used in cancer in many, many places. I mean, in many ways here, for example, the Netherlands and the US are further ahead than the UK, for example. And that's all credit to the systems in the Netherlands and in the US. But it's a wonderful opportunity for producing better diagnoses. And then the final point that I think we'll end up with genomics is when we do this across the entire population to inform lots and lots of health decisions as part of your medical record. People have been talking about this for ages. It gets closer and closer and closer every year, decade, 5 years or something. I think a particularly interesting country to watch here is Estonia, with the Estonian Biobank, where they have about 1/5 of their population in their research cohort, and they're very likely to end up with kind of stretching to their about 1.2 million size population as a whole. Denmark and Finland are also really interesting to watch. And then the UK's newborn screening and Our Future Health are two newborn screening from Genomics England. And this new project called Our Future Health makes the UK also interesting here. And how we use it will be interesting. And I think it will be used in a rather unsexy way at the start. So not in a kind of, I don't know, I'm going to tell you everything about you because of your DNA. That's not true, by the way. Our DNA doesn't sort of define us, and you can't, age 0 tell you whether, when you're going to die or how good you're going to be at some sport or something like that. It's just not the way it works. But what I think we will be able to do is like tweak and improve our screening criteria, tweak and improve how we do, for example, statin prescriptions or breast cancer screening or colon cancer screening or these other things. And so we'll be making just a slightly better screening system. Now that doesn't sound very sexy, but it has a really big impact when it's done well. And so we're going to have to like crunch the numbers. A number of people are crunching the numbers well here to like steadily improve how we look at some of these chronic diseases.
Great. Lastly then, any keywords of advice or lessons that you'd want to share with early career researchers in genomics and/or personalized medicine?
So for the clinicians who are watching this, channel that inner X-ray story. In fact, I would read up on the history of medicine about images and X-ray. It's a great story. It involves Rotterdam. It involves fairgrounds in America. It involves the First World War and tuberculosis. So it's a great history of medicine around imaging. And for genomics researchers, welcome. We need you. Please stick with that. And if you have not learned how to code, learn how to code just a little bit. So you will be so surprised how a little bit of Unix and a little bit of Python and a little bit of R will transform your ability to do data science. You do not need to be like some geeky person to do this. Just a little bit, going one step beyond Excel, go one step beyond Excel into Unix, bit of Python, bit of R will really, really help you.
Brilliant. Thank you so much for your time. It was great talking.
My pleasure.
Thank you.